Hi, I'll be talking about how host race formation is maintained in a vector-borne parasitic plant system. All right. So genetic differentiation following the colonization of a novel host species is hypothesized to be one of the major factors influencing the rampant diversification of many parasite lineages. But in order for this so-called host race formation to be maintained and potentially lead to speciation, parasites need to be able to find the right mates, so ones of the same host race to exchange genetic material with, and they need to be able to disperse to the right host species. Vector transmission is required by many parasites, and it can complicate adaptation and subsequent diversification to a host species. So a parasite that is vector transmitted is somewhat at the whim of a vector when it comes to who they're going to be able to mate with and who they're going to be able to infect. So I study how vector transmission is affecting the process of host race formation in a system where we can follow vectors and parasite propagules in the field. This is desert mistletoe, so it is a dioecious hemiparasitic plant native to the Sonoran Desert that infects leguminous trees and shrubs. Today I'll just be focusing on two of the hosts, velvet mesquite and catclaw acacia. So in order to reproduce, desert mistletoe requires a vector of its pollen, and this can be any of a number of generalist insects. And then once pollinated, its seeds must be dispersed primarily by a relatively specialized seed dispersing bird, the phainopepla. So I won't go into this today, but neither of these vectors is specific in its activity to parasites of a given host. So we might expect their behaviors to erode any genetic differentiation that's being built up by, say, the parasite's developing ability to get around, resistance from the host themselves. So even though we see in reciprocal transplant experiments that there is reduced success of seeds transmitted between hosts, these vectors, their non-specificity might be eroding that differentiation through time. However, we see in the field over the spatial extent of just a few meters, if we look at the population genetic structure of mistletoes on acacias and mesquites, we see strong evidence of genetic clusters associated with host species. So here I'm showing a structure plot where each vertical bar along the x-axis represents an individual whose ancestry, as measured through a number of microsatellite markers, is partitioned among these two genetic clusters. So we see the strong host race formation, but we don't see very many individuals that are the result of crosses between the two host races, and so therefore due to pollination. And we think the mechanism for this is because of differences in the flowering phenology of the two host races. And we see some evidence for this flowering phenology being under partial genetic control, and not just simply a response to the physiology of the hosts. In contrast, we see a handful of individuals that appear to be the results of successful dispersal events between the host species. And this is consistent with the fact that the fruiting phenology is constrained regardless of host species, and we think it's constrained to overlap with the specialized seed disperser. So in general, we see that dispersal has less of a potential to provide a barrier to gene flow between these host races than pollination does, but current work is looking at what the origin of this pollination barrier to gene flow is and whether it's being reinforced by selection. So with that, I just want to thank the people and the money that made this possible.